of Alzheimer's disease, and you will also have an increased risk of leukemia. So now, our lab has been trying to understand how chromosome 21 in human actually leads to all these problems that affecting Down syndrome individual. And one of the characteristics that we have a lot of interest in is, in fact, the neurogenic to gliogenic shift. And what does it mean? People with Down syndrome, they have less neuron at the time of birth. And on top of that, besides having less neuron, they also have more astrocyte. I think this is the most important key. If you don't remember anything today after this lecture, remember, Down syndrome individual has less neuron to begin with, and they have more astrocyte. And as you know, neuron is the key to our intellectual capability. And not just the number of neurons, it was also the connectivity, the connections that these neurons make that actually plays a huge role. Remember that. So what are we going to do is, people with Down syndrome basically have less neuron, and we try to understand what is actually going on with that, what actually makes the cell in our brain decide to turn into something else but not neuron. So the one that we have been looking at is by using this mouse model that we have, and this mouse model has Down syndrome. And of course, it doesn't have all the characteristics featured by the Down syndrome individual, but it basically has the most important features that we are looking at, which is the learning and memory defect. So this mouse model could not learn and could not actually remember what they have actually learned. And besides that, this mouse model also developed all kind of pathophysiology that also similar to the human individual with Down syndrome. And therefore, in the past 10 years, we have been studying this mouse model. And what we have been looking and seeing is these cells in our brain called progenitor cells, or maybe make it simple, a baby cell. This baby cell, just like any one of you, right? Before you come and join this university in the program that you like, so you have to make a decision. So this baby cell decide to turn into something else but not neuron. And what we have actually seen, it basically produces less neuron. It doesn't want to become a neuron, and they turn into astrocyte. So cut it short. So what have we done is we cut open this brain in the mouse model, and we look into the most important part of the brain, the three major parts: the olfactory bulb, the cerebral cortex, the cerebellum, and the hippocampus. These are the regions that basically control the learning and memory, motor coordination, as well as long-term potentiation. So what we would like to find out is what exactly may have gone wrong to this region. And we basically used microarray back then to understand what could have actually gone wrong at the genetics level. In terms of what gene is being expressed more and what gene is being expressed less. So to cut the story short, so we basically perform a microarray on 72 samples from all the different regions, different time points between the two groups, the mouse model for Down syndrome versus a normal mice. So what have we found? We found one particular pathway known as JAK-STAT and interferon signaling pathway happens to appear all the time in all the different time points throughout development in this mouse model. And what exactly is JAK-STAT doing? So first, we try to understand, does this particular JAK-STAT, it's just a gene, okay? Is this JAK-STAT expressed in the brain? And we look into the different type of JAK-STAT. Of course, we have many types of JAK-STAT, and we look into different type of JAK and STAT, and we look into all the different time points and different brain regions in the mouse as well as a human. And it happens that JAK-STAT is in fact very important. It happens to be expressed in the brain all the time. And if you don't have JAK-STAT, you will be in big trouble. So that is the bottom line of it. So what exactly JAK-STAT do? So remember the story I was telling you. You have less neuron, more astrocyte. And your baby cell always make a wrong decision in Down syndrome individual. So what happened is, JAK-STAT is increased. You have a lot of JAK-STAT in Down syndrome individual brain. So what has happened here is, if you have a lot of JAK-STAT basically help your baby cell and tell the baby cell, don't turn into neuron. Okay? So instead of asking the baby cell don't turn into neuron, the JAK-STAT basically ask the baby cell turn into astrocyte. And we believe this is the reasons why Down syndrome individual has less neuron and more astrocyte. Okay? So what could have happened if a Down syndrome individual? Why do we think that is the case? Okay, one of the very important thing is just that works closely with the interferon pathway. And what happened is that you can see the one on the left, on the right, sorry, on the my right, is actually the dystrophic figures. This is what happened in normal mice. The one on the left is in fact what is happening in the brain for the Down syndrome mice. What happened is you have a lot of these windows. Just imagine you have a house with a lot of windows.